Greer. He's an assistant research professor in the Division of Marine Science at the University of Southern Mississippi here, working for the Concords Consortium. His research interest includes zooplankton and larval fish ecology, planktonic food webs, plankton patch dynamics, and biological sampling technology, particularly imaging systems and acoustics. And he's going to go ahead and share his recent research with us. Thank you.
Well, thanks everyone for being here today, and thanks to Monica and all the organizers for inviting me. It's an honor to be here. I'm going to be talking about this system that you see on the screen that I've actually been working with for about 10 years now. Time flies. All these different images you see here were taken with this system in the Gulf of Mexico. And in particular, I'm interested in plankton and how they may interact with oil. So just to get everyone on the same page, as you all know, the Deepwater Horizon oil spill happened about 8 years ago. 11 lives were lost. It was a huge chaotic scene at the surface. And what Guillaume just went over, you can kind of see in that picture on the left side there, how immediately after the spill, the ocean currents were taking over, aggregating the oil at the surface, moving it around. And this was further complicated by the application of dispersants at the surface as well. And so there's the big hot topic of research is just understanding where the oil went and how it changed with the dispersants and other things going on in the area. Below the surface, the situation is also very complicated. We had dispersants being applied directly at the wellhead, and then a pretty well described deep water plume at about 1,100 meters, where you had trapped oil and gas. And this figure is showing kind of these little particles raining out. This is the marine oil snow that took material from that trapped plume and exported it back into the sediments. And about half of the oil made it to the surface, where we saw a lot of the immediate environmental effects, dead seabirds, whales, very obvious environmental problems to us. But one of the big questions was how some of these smaller, more abundant animals were being affected by this oil. And these are very important parts of the food web, but it's a lot harder to see the effects and understand how they're going to propagate throughout the ecosystem. And so that's really what I'm going to talk about today, are these planktonic animals and how we're using this new technology to understand what happens under normal circumstances so we can better understand what would happen in an oil spill scenario. So what is plankton? One of the that gives you a key or an idea of how it lives is the word plankton, which comes from this Greek word meaning wanderer or drifter. So these are animals or plant cells that cannot swim against the prevailing currents. So they are at the mercy of the currents. There's a bit of ability to migrate vertically, but they cannot swim against your average current in the ocean. Three groups. We have the phytoplankton. These are kind of the powerhouse of ocean life. They are taking carbon dioxide and converting it into usable compounds that all the other animals in the ocean rely on. In addition, as a waste product of this photosynthesis, they produce oxygen. Approximately half the oxygen in our atmosphere is produced by phytoplankton. The zooplankton are all the animal plankton, and then they have this kind of catch-all category of marine snow, a very hot topic in a lot of the oil spill research. That is essentially detrital, dead or decaying material that sinks through the water column that has biological origin. So phytoplankton, here's an image of phytoplankton. You can see these hair-like, essentially chains of phytoplankton cells. These are diatoms. It's one of the most important phytoplankton globally. The zooplankton are divided up into two groups. You have meroplankton and holoplankton. So meroplankton spend a portion of their life cycle drifting in the currents, and then they eventually grow to a certain size where they become nectonic. They can actually swim, or they settle in a benthic or bottom habitat. And this includes the larval stages of almost every fish imaginable other than sharks and rays. They have tiny planktonic eggs that they spawn in the water column. Those hatch into larval forms of various kinds. This is a pipefish. You can see that narrow thing. There's a tail on the top and a little snout on the bottom. You have a flatfish. This one on the left middle is a lobster larva. The two on the bottom are different stages of crab larvae. And this squid-like thing is actually a tube anemone larva. And so there's just a bunch of different larval forms that are out there all drifting around in the currents. The holoplankton spend their entire lives as plankton. These include copepods on the far left top. Those are one of the most important consumers globally. These three groups on the top half are consumers of phytoplankton, and the ones on the bottom are consuming other zooplankton. And the marine snow, these are essentially dead or decaying material, including exoskeleton. That's a shrimp exoskeleton you can see sinking through. And so there are really a lot of different forms. The plankton are actually quite beautiful, as you can see, but scientifically they're interesting because they're this critical link in the food web. And not much is known, especially in the Gulf, about different processes and why certain groups show up where at different times. In addition, like I mentioned earlier, the early life stage of many marine animals are meroplankton, and so we have to understand some of the oceanographic processes so we can better understand population changes for all these different fish species. They're also, because they have really fast generation times, they're essentially a canary in the coal mine. They can tell us that they have a biological indicator of any stress on the ecosystem because they can rapidly die off, they can rapidly respond and grow faster under different ocean conditions. And marine snow is this hot topic in the oil spill science because it can export carbon, so it's part of this biological pump in the ocean, but it can also entrain other materials such as oil and pollutants and then move that around. So one way that we can look at the distribution of plankton is with satellites, which we mentioned earlier. Brooke's going to talk more about remote sensing. But so in this image here, it's a map of the northern Gulf of Mexico, and the warmer colors essentially indicate that there are more phytoplankton in that area near the surface, and the cooler colors indicate lower abundance. And so if you look at where Deepwater Horizon was, it was well offshore in deep water, not a whole lot of life out there relative to the coast. And I don't want to say that there's nothing out there that's of interest, because there's certainly many important habitats, deep water habitats, that are really understudied. We just don't know a whole lot about them. But if you want a concentration of life, the coastal area is where there's a lot of fisheries, there's a lot of plankton. It's just where life tends to aggregate. And so when we want to think about oil spill impacts, certainly what happened at the site is important, but it's also important to know what's happening on these how the oil is getting to these coastal areas and what's happening once it's there. And so as part of my work, I'm in the Concord Consortium, which is studying this nearshore habitat, which is a critical area for fisheries. And also there's a lot of freshwater input in this area, such as river plumes. There are fronts and these very ephemeral features that show up that are very important for aggregating plankton and also oil. And so plankton ecology, in my view, it kind of links these two topics, because you have organisms that are being dispersed by the prevailing currents, but then it's also this critical link to fisheries. And so it's a really interesting topic that technology is helping us understand quite a bit. So to understand the importance of this new technology, it's important to kind of take a step back and understand how we sampled plankton in the past. And what we still do today, actually, is use this technique called a bongo net, which is essentially a net that we just tow vertically through the water column, and you get one big sample. But the problem is you have no idea where in the water column that sample came from. So if you were to want to understand the exposure rates of different plankton, you want to know if they're at the top of the water or the bottom based on where the oil is. And so people realized that this was a problem, that you're just averaging across the entire water column. And so they developed these multiple opening and closing nets that allow you to get vertical bins where you have different samples in different parts of the water column. But you're still limited because you can't really open them every meter. Say if you have a freshwater plume that's very small scale, you can't really capture a lot of the biological variability around that feature. There's also some really severe biases with these net systems for organism detection. So anything that's slightly fragile, that doesn't have a hard exoskeleton like a shrimp or a copepod, is going to get destroyed because it gets pinned up against the mesh and just gets obliterated essentially. So all these gelatinous organisms that we know are very important because there's a ton out there just aren't quantitatively described with a lot of these systems. And so with the advent of more computing power in the 1990s, we started to see kind of an explosion of different optical systems that were developed, including some at Woods Hole. The video plankton recorder on the left was developed at Woods Hole. And this also the one on the right was developed at Woods Hole. And then there's the one in the middle is pretty popular in Europe still, underwater video profiler. And so all these systems, they have different optical techniques. They're all sampling at a very high resolution, so you get really good vertical resolution of where different plankton are aggregating and things like that. There's also this potential that's a really hot research topic, is trying to take the image data and automatically process it and get biological information much faster than we do with traditional net-based sampling. And there are also fewer biases among different organisms. The real difference between the systems are the volume that they sample, and so if you're after some rarer plankton, you generally have to sample a larger volume. So the video plankton recorder, for example, measures a pretty small volume. It's great for looking at copepod abundances because they're very abundant. But the system I'm going to talk about today is the in situ ichthyoplankton imaging system, which falls under the large volume category. And so we call this thing actually the ISIS, which people talked about that name before ISIS was a terrorist group, so I don't want to change it because then they win. So so the way it works, so this is the system right here, and so the bottom two pods are where the imaging happens. And this view, it's like an overhead view of those two pods on the bottom. So you have an LED going through a pinhole, and it hits a plano-convex lens, and then a mirror, and it gets projected across this area, which is where the water is. The imaged water parcel hits another mirror and then goes into a camera. And so what happens is that whenever there are plankton or particles in this area, they actually block the light source, and so we image them as shadows. And this is called shadow graph imaging. The advantage of it is that when you backlight something, you have a large depth of field. So anything in this imaged water parcel stays in focus, and so you can image a relatively large volume of water. So we use a line scan camera, which is good for towing. And then it has motor actuated wings that you can see on the side here. These actually, we have software on board that allows us to change the angle of the wings, and so you can allow the vehicle to dive up and down through the water column while taking images. And we collect a lot of data, about 2 terabytes of images collected every 3.5 hours of towing. And so this is just kind of how it looks when we tow it behind a ship. It's on a tether that basically beams up a bunch of image data along with salinity, temperature, depth, barometry, proxy for phytoplankton abundance, dissolved oxygen. And so we get this massive data set, a bunch of files that are images and a bunch of physical data, we call it, and we can merge those based off a common timestamp. So we get an environmental information about every image that we take. And so for the Concord Consortium, we actually sampled with this system along these 3 pink transects. You can see that so we're confined to the coast, and we're essentially trying to describe the distribution of these different plankton groups amongst 3 different seasons. So what did we want to get out of this technology? We want to look at the physical structure, essentially the temperature distribution, salinity, all those things, detect different features that may aggregate plankton or affect their distribution, such as fronts, eddies. These are all very important for biological productivity in the ocean, and couple these with known current structure. And then we get biological structure. We can look at a lot of different information from the images. And the key thing is that your physical data and your biological data are on the exact same scale. So it's a really good instrument for looking at how the physical environment is structuring some of the abundances of different plankton. And so this is relevant to ocean ecology just in general because we don't know much about the distributions of plankton on fine scales, and it allows us to kind of get an understanding of if there were hypothetically an oil spill, which groups would be most exposed, what would be the rate of exposure. And so the data kind of look like this. I'm just going to kind of orient you to this graph. The y-axis is depth, and the x-axis is latitude. The color is the salinity of the ocean in that particular area, And the size of these rings corresponds to the abundance of these two different groups. So the larger the ring, the more plankton there are of that particular group. These are in individuals per meter cubed. And so these are two gelatinous organisms that have dramatically different distributions. You have doliolid, which is a phytoplankton grazer. It's consuming phytoplankton, aggregating along the picnic line here, or the halic line, and then a lot of it underneath. And then this is a ctenophore, which is consuming copepods, and it tends to be in the top part of the water column, especially in these two areas of kind of intermediate salinity values. And so you can imagine if there were an oil spill, the exposure rates between these two groups would be dramatically different depending on where that oil was. And so one of the topics that I've been researching for quite a while are plankton thin layers, which are dense aggregations of plankton, usually several times the background concentration, where they just essentially aggregate in an area less than 5 meters vertically. And basically nothing is studied about these in the Gulf of Mexico. However, there are other environments where they're very well described. The ecological significance of thin layers is that they provide kind of this concentrated feeding zone. So you can imagine two scenarios here that I've outlined where you have a jellyfish predator and then a bunch of copepods that it's eating. In the two situations, you have one random distribution and one where they're aggregated in this thin layer. And so the predator can exploit the prey resources much more rapidly when they're aggregated in that thin layer. And so they're kind of this trope. They consider these a hot spot of a lot of activity when you see these layers in different systems. And then, of course, if these layers are very prevalent, it can affect how we understand exposure rates of oil. And so this is going to give you, I'm going to show a video of one of these tows up through the thin layer. And so this is an image. Essentially, this is kind of what you would see if you were on the ship looking at a live feed. And so a lot of these little hair-like things are diatoms. Some of the smaller dots are copepods. The green dot is here, and it's going to move across the screen. It's going to show you exactly where you are in relation to that layer. This graph is showing, the top graph is showing chlorophyll A, which is a proxy for phytoplankton abundance, and you'll see that it's very accurately representing what's in the screen. And then we have salinity and temperature on these two graphs. And so I'll just play it. And so you can see just how dramatically the environment changes as you go up into this layer. You'll start to see these really thick diatom chains showing up. And then there's a lot of these barrel-like organisms that are doliolids, and that are phytoplankton grazers that are aggregating in that layer as well. And so it's very brief, but it's a just massive aggregation of plankton within a very confined area. And then as we get further above, you see the water columns start to clear out a bit, but still pretty abundant on the doliolids. And so that was detected in the western side of our sampling area, kind of towards the 25-meter isobath or so. It's a pretty dramatic feature. I've actually seen a lot of thin layers in different environments, never seen one quite that dense and that confined to such a small space. And so if we look at the abundance of the different plankton around this thin layer, so this graph is showing the chlorophyll A, which is the color. So the red is where the thin layer is, essentially the lighter or the hot, warmer colors. And you can see these are chaetodnasts, which are arrowworms, and shrimp. And so chaetodnasts tend to be aggregating at the surface and also right after the layer, in similar pattern to the shrimps. But then when we look at hydromedusae, which I show here, they have quite a different distribution where they're actually deeper in the water column and confined to the, so the left side of the graph is towards the north and the right side is to the south. So they're just north of the thin layer, and then they kind of become less abundant. And these are the doliolids, which you saw in the video. This is the oxygen distribution. So you can see the thin layer, you have very high oxygen, which is not surprising because they're photosynthesizing, producing a lot of oxygen. And these grazers tend to follow the trajectory of the thin layer, but they're not really in the most shallow part of the water column. So you can imagine just very different distributions for similarly sized organisms occupying these different areas of the water column. So just to take-home message is that a lot of these imaging data can reveal some of these key relationships between the abundance of different groups and the oceanographic properties. It kind of sets the stage for these other questions about, so if you were to able to simulate the distribution or trajectory of oil, what is the biological setting that that oil is going to be propagated around through? So we need this information to kind of understand oil exposure rates in the Gulf of Mexico. And also, kind of as another take-home message, the ICES data are really complex. And so an image is worth 1,000 words, as they say. It's also it's hard to quantify exactly what you're seeing in these images. And this is kind of where there's a lot of research in artificial intelligence and things like that, where people are working on a lot of these fundamental problems about how you can say that this, for example, this anemone larva is consuming a salp down here. How do you automatically pull that information out of these images? And so it's kind of this, and we wrote a paper recently about how a lot of these lobster larvae are attached to gelatinous zooplankton. And so there are these neat behavioral interactions that no one's ever seen before because we've been towing nets through the water for about 100 years. There's also some interesting behaviors of the larvae where they curl up in a little ball. This is an eel larva that's all curled up, and it's hard to understand what's happening unless you've seen a lot of these images before. And this is a flatfish larva that's facing into the camera. And so there's just a lot. It's a hot research topic of how we can understand and get the most out of these images automatically. And so with that, I'll conclude and thank you for listening.